assignment and everything, all the details that you need. If you find something unclear or anything, let me know. It's easy to update. Also today, what is it? I'm going to upload. Why is it not here? Okay, I also uploaded this list of papers that we're going to present through the course. And once I know the list of the students, I will just assign more or less randomly the people to the papers, but it's not like a Catholic marriage. If you want to switch or swap, just do it between yourself. It's kind of, I don't want to get into that. Only let me know if you have some issues with that. And
so you will random assign students to present the paper?
I have not yet, but I will.
Okay.
Okay, and I will just kind of write a name on that once I know who's presenting. And also today, if I'm not mistaken, I'm going to upload the first assignment, which is another document, quite detailed. And the idea is that I mean, don't take too much notice now because the papers are going to change. But the idea is that you pretend to be now someone who's approached by an editor of a journal who received a paper for publication, and they ask you for your opinion, critical review of the paper. So I give you an option between 2 or 3 papers to choose from, and then I basically explain to you the process, what the editor expects you to do, what are the things that you should pay attention to, and I also give you like guideline questions, so things that you might kind of have in your head when you are writing the review. And at the end of the document, I give you a review that I copied. It's not written by me, but it's like a paper that I reviewed, and when you review a paper, usually you get also the other reviewers' comments. So I copied the comments of another reviewer on this paper, and you can see it's a long review. I only let you do 2 pages, but this is a much longer review where the reviewer addressed many points, and I put it here so you can kind of see the style and how you write your argument when you want to convey a message to the editor. So you can kind of go through these not in too many details, but just to kind of get the feeling of it, how it is actually being done.
When is the submission time?
So all the deadlines for the submissions are written also in the syllabus. And usually I give you 3 weeks for each one of these assignments, and then 1 week break between the submission and the next assignment. Okay, and I remind you that you're doing it in pairs, and yes, this means that I will have to check 12 assignments. Okay, sorry, so we're going back to our topic. And I'll just give you a quick reminder that we talked about the coding of in the neural systems. How? Sorry, why is it not on? Okay. Are there any protocols that the nervous system is using? What are the meanings of the spikes? And we talked about different ways to encode data, to encode information, and also 2 options to go from top down or bottom up. And we looked at some recordings of spike and the spike interval distributions. We talked about the idea of what is a Poisson process and what are Poisson neurons, and what are the possible sources of noise. Okay, and we discussed that paper by Main and Chesnovsky, and I think we ended up by talking about this very specific case where the nervous system basically invented a system which allows it to be more accurate in the detection of individual spikes, and the resolution is a tenth of the width of the spike. So you get two spikes from different ears, and still the system can detect the difference between these two spikes, even if the difference is the width of a tenth of a spike. And the system goes, I haven't told you that before, but the system goes to really high, making a lot of effort to do that. And one of the things that you can see in the system is this very famous synapse between two neurons, which is called the calyx of Held. Okay, calyx of Held. And this synapse is, you're getting a presynaptic axon and a postsynaptic neuron, and you see this neuron almost doesn't have dendrites, and the synapse, it's a huge synapse. It goes over the neuron, hugs the neuron, and makes something like 200 or more contacts, synaptic contacts between the presynaptic axon and the postsynaptic neuron, which means that every time you get a presynaptic action potential, you always get a postsynaptic action potential. It's basically a relay, one-to-one relay between the presynaptic axon and the postsynaptic neuron, and it is very famous because this is the only system in the brain where you can actually patch the presynaptic terminal in mammalian brain, where you can patch the presynaptic terminal and the postsynaptic neuron, so you can actually see what happened in the presynaptic terminal and the postsynaptic site, and can measure things that otherwise are too difficult to measure, because usually this would be about 1 micron size, and this would be a spine, and there's no way to address them electrically. Question? No, this is a chemical synapse. It just has many release sites. Okay. So a lot of work has been done, like voltage clamping the presynaptic terminal, postsynaptic terminal, and playing with all kinds of things. So a lot of what we know about synaptic transmission in mammalian brain is due to the synapse, and of course you have to understand that this is also create some biases, because this is a very special system, so it might not be very representative. Okay. So we basically saw that neurons could be, first of all, they are stochastic, but they are also reliable, and now the question is, there are two questions that we can ask. One of them is kind of strange, and I'll try to explain to you in a second. And the other one is, if they are stochastic, what is the source of the noise that we see in the system? So to explain to you what I mean by what are they hiding, and this is kind of, I'll give you an example. It's not like the best example, but I think it can explain things, especially to the ones. How many of you took the course of Tali, of Chishuviyut? So, and how many are not doing it right now as well? Okay. How many of you who don't take the course knows what entropy is? Okay. So here's an example. It's a simple example, and I assume it's kind of designed for biologists who are thinking a lot of DNA, but it's just kind of an example. So imagine that you have a sequence from the genome, and you know that it's made out of 4 letters, encoding some, say, protein. Okay, and now imagine that this is like part of a very long sequence and you want to transmit this sequence somewhere else. Okay? So one thing that you can notice if you look at this sequence is that the occurrence of certain letters happens more frequently than others. So C happens more than T, and G is quite rare. Okay? This means that the representation here is not extremely efficient because you repeat a lot of the C's, but you don't use G's very often, but they occupy the same space. Okay. So you can imagine that you want to represent the sequence in a different way, and one thing that you can do is assign for each one of the letters, okay, a code which is made out of zeros and ones. Okay, and this is a kind of an example code that if you learn with Tali, you know it's a Hoffman code, but that's not the important part. The idea is that now you see that the C is represented by 1 0, but the G is represented by 3 ones. Okay, so now if you write this sequence, it becomes, so it's of course it's longer because now you're using only 2 symbols compared to here that you used 4 symbols, but this sequence is now more, is shorter in terms of like if you just count every 2 as 1, then this, you can check later, that this sequence is shorter, and it still transmits exactly the same amount of information. Okay, so we managed to compress this sequence into a shorter sequence by using this representation of that depends on the frequency of the letters. So letters that are frequent, we are using shorter symbols to represent them, and letters that occurs rarely, we're using longer sequences to represent them, and we're doing it in some optimal way. Okay, but this this has 2 consequences. So this is the basis of information theory, and a lot of everything that is happening in terms of communication now in the world is based on this observation and some mathematical theories that are using it and optimizing it, but it fundamentally it has 2 properties that are always there and are very important. One is that you see that if we do this in an optimal way, you see that now the probability of the zeros and ones to happen is equal. Okay, they're happening in the same probability. And this process is called whitening. So the idea is that now there is no redundant frequencies. I mean, there is no symbol that appears more than others, so you cannot compress it anymore. Okay? So when you get to the ideal situation, the probability of each one of the symbols is random. It's more than that. It's also if you would look like what's the probability of 1 to appear after 0 or 0 after 1, all these probabilities will be, if you did it like in the optimal way, all these probabilities would be equal. Which means that if you don't know this table of the code, so everybody is with me now, or I'm completely lost? If anyone wants me to repeat something, let me know. Yes? So if you count it here, I don't remember, but like, okay, 2, 4. Yes, but here you are using 4 letters. So now if you, and suppose you represent each one of them with 2 symbols, then it will be longer. It will be 32, right? Okay? Okay? So what I was saying is that when the code is optimal, when you compress the signal in an optimal way, the occurrence of each one of the symbols is random. Which means that if you don't know the code that you used in order to generate this, it will look a completely random sequence. Okay? You don't know. It looks like someone was just flipping coins, and you don't even know that this represents any information at all. Okay? So So this process is called whitening, and this is a result of being, doing a perfect whitening, that you don't have, that when you look at a signal and you don't know that it's coding something, it looks completely random. Okay. There is another consequence which is less important for us, and it is that these codes are very susceptible. So if I flip by mistake, when I transmit it on a line to a different location, and some of the bits would be flipped between zeros and one, then the whole thing will break down when you'll try to decode it on the other side. Okay, so if you take this table now, and I flip here 0 to 1, this 0 to 1, and you now use this table backwards in order to reconstruct the thing, the whole thing will break down. Okay, it will not be this sequence. Some of the letter would flip, and it can propagate for a long time until you get back to the original sequence, and this is very bad. Okay, in genetics, it's exactly the same problem, right? You get mutations, and everything gets screwed up. So it's the same problem. So, but one of the reasons that you have some redundancies is that you can kind of correct some of these errors. When you completely compress the thing and make it completely random, then you cannot correct for it. It's very difficult or impossible to correct for it. So there's a whole theory for that, and it's not part of our course. But the intuition that I'm trying to convey you here is what went through some people's minds when they saw the things that we saw here. Okay, so they were thinking, okay, we see these Poisson processes. They are highly random. Basically, they are the most random that they can get. Maybe this means that the code that the neural system is using is like a very, very efficient code. It's not representing real noise. It's just representing information, but we don't know the code. Okay, this is what I'm trying to say when I'm saying, what do the neurons hiding? So you can expect that if you tap into a system and you see things that looks completely random or nearly random, this means that what you see is a compressed signal, is a signal that has been optimized for transfer of information, and it is encoded, and there is a code, and if you don't know the code, it looks random. Okay?
Are there any evidence for a code?
This is kind of an evidence. Okay. That's what we're trying to figure out now. So this is kind of white of some of the feelings. Okay. So this is one intuition about this variability. So the kind of the question that's hanging in the air is: are the neurons random, or are they reliable? And the variability represents something else. For example, it represents some underlying code that we don't understand. Okay, that's one possibility. But here are some, it's kind of, information theory is a very well-known and very established theory. Everything is very clear there. Here, it's what I'm trying to tell you, it's like bits and pieces. It's not completely round story. So I can't kind of connect all the dots. No one still can connect all the dots. But there are open questions around these things. So I'm trying to give you bits and pieces about these questions. So here is another kind of approach toward the same problem. So imagine that you have a neuron, and you know that neurons receives a lot of inputs. We talked about it before, and we talked about this idea of averaging the inputs. So Moshe Abeles, who was a scientist here in Hadassah, and he was one of the founders of ELSEC, he was doing many recordings in the auditory systems of cats, and then in the premotor areas in monkeys. And one of the things he came out with, the idea was, imagine that you have this neuron, and it receives a lot of input, Uh, so the neuron, if it does what we think it does, which is summing the input, so taking taking the synaptic input, making a summation of the input, and then when the sum reaches threshold, the neuron would spike. If you would do that, you can do a simulation like that. Just generate like random spikes. Okay, take the sum of the spikes and then run them through some kind of a neuron that makes an integration of the neuron. What you will see that happens is that because of the laws of large numbers, the central limit theorem, you basically get just the average. And so you get an average kind which is more or less constant, which depolarizes the neuron. You get to a spike, and then it resets, and then it goes again, and it resets, and you get a very, very regular output spike train, even though the input looks completely random. Okay, just because the neuron basically just what it cares about is the average number of input that arrives to it, and this average number is more or less constant all the time. Okay, everyone is with me? Okay.
But this is for uniform weight.
Uniform weight.
Right.
But if you, yes, but it doesn't, so the whole thing, it's true, but if you get non-uniform weights, it just scales the number somehow. So you might get some version of, a regime where you can kind of change somewhat this regularity a little bit. But it's very difficult to get from here, even with non-uniform weights, to something that is irregular in the output, like we saw before. But we'll talk about it more, Neil. There are, yes.
Why, I mean, I understand that if you see this, it looks like the input is random, but
Why would we assume that the input is random? So this is kind of it's a thought experiment, if you want. Okay, you put a neuron, you put an electrode in the brain, and you see this non-regular firing rate that we saw. Okay, and you ask yourself, okay, I have like many neurons. He basically wrote a whole book about it, which is very highly recommended, called Corticonics, and you are welcome to read it. But the idea is it's really a thought experiment. You put an electrode, you see this random inputs, random spikes of output. Now you say, okay, I take one neuron, it receives a lot of these inputs, okay, but this neuron needs to be consistent. It also needs to be spiking irregularly. But if I would just kind of feed them and do with the neuron what I know that neurons are doing, what I will get is actually that. And you can actually do a computer simulation and show it. Okay. So he said, okay, but let's assume that this is not what really happens. Let's assume that the input is really like that, but now what the neuron cares about is not, it's not like summing all the spikes with like very, very little EPSPs each one, but actually what the neuron cares about is what happens in really very short time beats. So the neuron doesn't look for the average input, but we just for a second we assume that the neuron would care for when does it happen that 5 of the inputs are spiking at the same time. Okay, so now what we're doing, we're taking bins over the whole thing here and count how many spikes happens in a short time bin. And now because this is short and these are random, we get something that is much more fluctuating. Okay, and then this turns out to be something that looks like that, which is much more random. Okay, now what's the reason for thinking that the neuron would do that? So you can think of, so this depends on a property of the neuron. What is a property that we are talking about?
Summation in time.
Summation in time, which is, how do we call it?
Time constant.
The time constant, okay, the RC of the membrane, okay? And for other reasons that I will explain to you later, we have reasons to believe that the time constant might be much shorter than we usually expect. Okay, question.
I'm not sure I understand the difference. In the first place, you just sum along the whole time axis, so of course it doesn't make sense, no? Because the second thing is just the same. That's what I was trying to ask before. Like, you still compute this like average or correlation of the inputs to the weights, or however you want to call it, but you do it in reasonable like windows. If I would do it to, I don't know, even without dynamics, if I would do discretize time to small enough bins and I would do the same calculation, I would get something that looks like the bottom figure and not. If I would just average, like multiply the input by the weight and then pass it through some mechanics.
So what you're trying to say is that the difference between here and there is the length of the time constant. What is a relevant time constant that the neuron actually is using in order to integrate its input? Okay, and I agree with you. Okay, so the question is basically do input. So I mean the conclusion here is if neurons integrate their input over 50, 100 milliseconds, then we have an inconsistency. Neurons get a random input and should output regular output, okay? But if they are integrating over 5, 7 millisecond time window, maybe then we will get something which is more consistent, okay? So this is a prediction, okay? This kind of, we have an explanation. Now we have a question, which is what is the relative time integration of input in the brain? Okay, let's put this question on the side. I'm not going to give you the answer right now, but we'll discuss it over the next few lectures. Okay. But he came out with so this actually developed into a model which is called the thin fire chain model. How many of you heard about the thin fire chain model? Okay. So the idea with the thin fire chain is the following. Imagine that the integration time window of every neuron is rather small, and neurons, in order to fire, they need many inputs in a short time being. Okay, so now imagine that you have a population of neurons, and they don't have to be ordered. So the idea, okay, let's imagine in the beginning that we actually have layers of inputs. Okay, you can forget about what you know about anatomy, just think about deep networks or whatever artificial neural networks that you have. And imagine now that these neurons are connected to a different group of neurons that are connected to a different group of neurons, and so on and so forth. Okay? And each one of these postsynaptic neurons shares some common input. So like this neuron, that neurons are connected to that neuron. Okay, and some of the other neurons are connected to that neuron, and so on and so forth. Okay. And now, for some reason, and this goes, in all layers. So now, for some reason, I activate all these neurons together at one time point. So the spikes goes to the postsynaptic neuron, okay, and some of these neurons will receive inputs that are happening together because these neurons were activated all together. Okay, these neurons will get this very narrow beam of spikes at a certain time, and they will spike together. So here, some of these neurons will also spike in a synchronized way. And the idea is that this wave of coincidence of spikes can propagate in the network. Now you can actually think about the geometry of this thing in a way that you don't really need this feed-forward propagation. You can actually think that this neuron belongs to that group, but it also belongs to that group. Okay, so it sends its axon to some postsynaptic neurons. They send the axon to some other postsynaptic neuron, and some of these neurons are sending back the inputs onto the same neuron. So the neuron can participate in different groups of this chain of events. Okay? And one of the things that I'm not going to go into details of, but you can read about it, is that there is this properties, kind of a physical or statistical property that you can think of, that if you have a process that has some variance, okay, and you look for, so imagine that these neurons are starting at some times, but they're not really precise completely exactly at the same time. They have some variance, okay? And now you look at this neuron that receives the input from them, and you look at when does it actually cross the threshold, and you know, it summates some of these inputs. Basically, the variance of these neurons in the time of when they will spike will be better, smaller than this one. So the kind of the precision is actually improves with time. It doesn't degrade. Okay, so you don't really need to activate all these neurons exactly at the same time. You can allow yourself some variance, and still the chain will maintain itself. Okay?
Do you have a length constant here, or?
Length constant in terms of what, the propagation delay
Yeah.
of the axons.
The upper neuron, does it activate, if it's activated together with the lower one, do they reach?
At the same time. Okay, so these are becoming like detailed questions, and it's true that, you know, we can't assume that the axons arrive exactly at the same time, and there are dendrites and there are synaptic delays. There are all kind of issues. So at the beginning, they were doing very, very simplified models like that, and then these models became more and more complicated, and there are, if you want, I can show you reference for papers where allow different delays between the this spike will arrive to this neuron a little bit later than it will arrive to that neuron, and still the whole thing works.
Then the variability will be larger.
Yes, but it's complicated. It's really it's a complicated thing, but you can look at it in different ways. If you try to design it, it's very complicated to allow for all this variability. But what you can look at it from the other way around, and you know, connect it randomly and see what happens. And then what happens is that there are always enough neurons. If you have enough neurons, you know, in every layer, there are always enough neurons to generate that will fire coincidentally, and then you know the chains maintain itself. And also, if you apply, you know, some rules of plasticity that I'm not going to, we'll talk about that at the end of the course, they can actually also correct all these issues. Okay? So this is thin fire chain. So this was one idea. And then came Sofsky. You remember this guy Sofsky that we talked about when we talked about the variability in V1, and he suggested a different way to look at it. And he assumed, and this is again, it's an assumption, it's not an experiment. He assumed that if the dendrites behave a little bit like axons, okay, not like what we know about dendrites. So imagine that in the dendrites you will have voltage-dependent sodium channels. So when you get an EPSP, nothing will happen. You'll just get an EPSP from one synapse. But if 2 or 3 inputs will arrive at the same location at the same time or very similar time, you'll get a dendritic spike. So you'll get a spike in the dendrite, like a spike in the axon, and this spike will propagate on the dendrite and will make the soma fire. Okay. If that happens, then we go back to this possibility, but now the time constant is becoming very, very short, like a 1 millisecond time constant of integration. Only so you have to kind of divide this into bins of 1 millisecond, and if it's enough of 3 spikes to arrive at the same location at the same time, we'll get a spike. Otherwise, no spike. Okay, so you assume that the threshold is very high for temporal summation. So you need many, many, many individual EPSPs to get to threshold. But if you get 2 of them at the same time, at the same location, you get a spike in the dendrite, and then you get a spike in the output. So Sofsky made this model, and his model, again, did something that was also very random. Okay. So again, with random inputs, specific type of integration of the neuron, we get random output. Okay? So now we have basically two ideas. Both of them are talking about coincidence detection. So we detect 2 spikes or 3 spikes or 10 spikes that arrive more or less at the same time in the input, and we can generate activity that looks random. Okay? And we have a self-consistent system. The system is getting random input and generates random output in some way.
But
Okay.
jet spikes only will active the dendrite also spikes?
Yeah, so now you have to assume that the dendrites have sodium channels and that they can do spikes. Okay, this is an assumption that we need to verify in experiments. Okay, this was in '92. Okay, that's a long time ago. But this was an assumption. This is a model. It's not real experiments. Okay, I want to just say one more thing. It's like we'll never finish this class. But one more thing about this, and it's not clear. I mean, what you see here is a self-consistent picture, but it's not clear where the information is stored, right? It's a strange coding system. It's not like what we think about neurons in V1. We show them a line, and they elevate their firing rate. Here, what you would assume that happens is that when the neuron gets, the right orientation, then a group of neurons is active, and then another group of the information is represented in a strange way, like waves that are traveling in the cortex or in the brain. And how do they represent information? It's not obvious. Okay. The other thing that comes out of this picture is that many of the spikes that are in the brain are completely irrelevant. Okay, so when you think about this neuron, it receives thousands of inputs, but only when very few of them are coincident together, then the input makes some output. So most of the spikes that are traveling and arriving to these neurons are just noise. They're not contributing anything to what the neuron is doing. Okay? So this means that not every spike is important. Only the spikes that arrives together at the same time to a certain neuron are important. This is a very different view from what we used to think about. Okay, so Sofsky published his paper, and then came a paper, very interesting paper, and there was like a back and forth papers arguing about this thing between two people called Shadlen and Newsom and Sofsky. And what they suggested is that both Abeles and Sofsky neglected something very important in this picture. Can any of you imagine what it was?
The shape of the dendrite?
No. Inhibition. I gave you the answer, but you have to read it. Okay, so if you assume that all these inputs are excitatory, then you get this picture. But if you assume that some of these inputs are actually inhibitory, then the summations become very random, no matter what your integration window is. Okay, you can do this exercise, but what happens is that you get like a random walk around some mean. Okay, and basically what determines now the output is a fluctuation around the mean and not the mean. The mean could be 0, so the neuron doesn't get any net current. If you just sum, the excitatory and inhibitory input, you'll get 0 current, and the neuron wouldn't do anything. But what happens is that the neuron fires because of the fluctuation around this mean, and then this fluctuation looks random. Okay, so you can generate exactly the same randomness in the output if you assume that you have input and out, excitatory and inhibitory neuron, out inputs, and you summate them and you get this random walk. Okay, so we basically created a problem that doesn't exist in a way. Okay, but in order for that to happen, you need to have some parameters matched. You need the excitatory and the inhibitory input to be somewhat balanced, because if it's not balanced, you still get this net current, and then you get this regular firing. Okay, so you need to get this balance between the excitation and inhibition. Okay, so people like Chaim and other physicists worked on this issue, and there are two famous papers that you probably talk about in neural networks, I hope. If not, you should put them somewhere in your reading list and make sure that you know at least what they're talking about. And the idea here is that when you have a network, you can take a network of neurons and connect them to each other, okay, by excitatory inputs between the excitatory cells, but also excitatory to inhibitory synapses, and inhibitory to inhibitory, inhibitory to excitatory. You have all these connections. And it turns out that in a certain regime, this network is in what they call the balanced state. So on average, each one of the neurons get a balance between the excitation and the inhibition. And when you run this network, what happens is that you get this kind of chaotic state. So what does it mean? So it means that, again, this is another way to look at randomness. That is, what I showed you before is from information point of view. You can think of things that looks random, even though they are deterministic. So they carry some information, you just don't know the code. Here you have, again, another deterministic process. So if you run the simulation, so this is an example where you run the simulation twice, exactly the same simulation of excitatory and inhibitory neurons. And each one of these neurons, I don't know if I have an example. Okay, each one of these neurons basically does something like that. Because it sums excitation and inhibition, it fires in a random way. Okay? So all of them looks very random. Okay? But now imagine that at some point in time, you take one spike out of the simulation and you force it not to spike. Okay? So you see that so what they do here is like the overlap, like what happened in the first simulation and the second simulation. And what happens is that the firing rate of each one of these neurons is completely the same, but the exact timing of each one of the spike is completely shuffled, and it happens very, very quickly. Okay? So immediately once you change one spike, everything is shuffled around. So here you have another way to look at it, where the system looks completely random, and it is random, but it is random in a deterministic way. So it's not stochastic, it's random because it's sensitive to very, very small perturbations. So it's enough that one spike is not happening and the whole thing looks very different. So this can kind of explain to you, so imagine that you are doing an experiment in visual cortex and you record from from a neuron in V1 and you present an input. So from this point of view, it's enough that one neuron on the way, and you have million neurons that are conveying information on the way, one neuron will not spike in trial 2, but is spiking in trial 1, and your neuron in V1 will give you a completely different output in terms of the individual spike. The rate would be the same, but the individual spike will be very random. Okay?
How do you see here that the spikes are shuffling?
So the kind of I think what happens here, I have to look at it again, but I think the idea is that this is, I think, taking from this paper. You take, you run the simulation and you do it in blue, and then you run the same simulation in red, and then you overlap what happened here after you removed one spike.
It doesn't seem like they are different.
So no, so now like each one of the dot doesn't overlap, so you don't see. So the change here in color is because here like all the reds are on the blue, but here suddenly the red and the blue doesn't overlap again.
The left also red?
Yeah, so the idea is that you kind of see something like that. So you have like the spikes in blue, whatever they are. Okay, and here you have the same. Just another blue. And like this would be exactly the same. And here, when you suddenly kind of miss a spike, suddenly everything is shuffled. So you have the same number of spikes, but they don't overlap, so the color is changing. Okay, because now you have both black and blue, and before you didn't see them.
They completely don't overlap, or they just
Some of them will overlap and some of them not, but like a lot of them will not overlap. And it all, of course, depends on a lot of the parameters of this simulation. I mean, what you put into this thing is determines a lot of what you get out, because this thing can be very deterministic, it can be very chaotic, and it can be on the border between them, so it depends where you are in terms of where you set the system to be.
Can this happen even without skipping the spike, like some ion channels, open regulator?
Exactly. So the point here is to assume everything is deterministic, and still you get that. Okay. Of course, we know that the system is not 100% deterministic. I mean, from basic physics, if you want, to what we know about ion channels, to what we know about synaptic transmission. So we have some noises in the system anyway. So it's impossible to completely repeat the thing. But what they want to show you here is that even if you could, the system is designed in such a way that it cannot exactly repeat itself 100%. Yes.
Does it matter if you shuffle at the
So this depends on what you care about. Okay, so now imagine that what you care about is the average firing rate of the neuron. Then in that case, it doesn't matter, because you don't really care when each one of the neurons happens, right? But if you have this model, then imagine that now one of these neurons doesn't spike. Okay, and then the whole thing falls apart, because you need to be exact on the spike. Okay, now it's not so easy, because you might design this system with inhibitory inputs in a regime where it wouldn't be that sensitive to a single spike, and then this system would be stable. Okay, with that model, yes, you could kind of take that model in a certain regime of parameters and make it with specific connections between. So people have done that. They took this type of model and they designed inside the network specific routes of connections, and then they show that they can actually propagate thin fire chains in these networks. Okay? But then the connections are not random. You have to build it in. No one managed to take a random network with this regime and generate that. If you do that, then usually they die out, these thin fire chains.
Is it possible to encode information
From here you can encode information on the average, in the average,
but
but it's not easy. I mean, only like 3 years ago, people managed to do, after 15 years working with this model, they managed to do orientation selectivity in V1 with these neurons. It's not obvious. It's very difficult, because these systems tend to basically just do noise, not anything else. They do what they want to do, not what you want them to do.
So what are the strengths of this model?
That it generates like a self-consistent model of the variability, if you want. But this is kind of an issue for neural networks. Okay, we can talk about it in the break. Yes.
Sorry, about the graphing the hope and graphing the touchdown. So it's slightly neural location in I mean, I don't know the difference between the hope and
What he
no, it's
here?
yeah. what's the
No, that's the population. It's like you look at what the exciter population doing and what the inhibitor population is
so
doing.
in the first graph it's all excitatory, in the second graph all
No. It's a one simulation, and you look at all the neurons. So these are the excitatory neurons. These are the inhibitory neurons. Every time you have a dot, one of these neurons spikes. Okay, so this is one neuron, this is a second neuron, third neuron. You have thousands of them here, thousands of them here. Okay, you start the simulation, the firing completely randomly. Okay, and you go to the end of the simulation, all of them are blue. Now you start the simulation again, and now you mark it with red every time the spike. But you start exactly the same, so every time the spikes happen exactly at the same time. Once you take one spike out, the whole thing shuffles out, and now you see the difference between the red and the blue spike.
so it's two simulations, but
No, it's it's two simulations, one on top of the other.
Yeah.
And
You just separate the excitatory and
So in this model, what determines the time when they start to overlap? Because
you repeat the simulation from the beginning. So
what parameters in the model determines the time?
When they start to divert,
Right.
or when they
start to overlap?
Start to overlap or start to divert? They overlap all the time.
I mean, start to divert, sorry.
The time when you take one of those neurons and you force it not to spike when it was supposed to spike. So in the first simulation it spiked, and now you don't let it spike at this time point, and then you let everything continue as from before. Okay, that's what makes the difference. Okay. Okay, so a lot of what we talked about here basically boils down to the question of what makes one neuron spike. Okay? We have to take a break. Okay, so I'll just go over this figure. And the question is, what makes a neuron spike? This is kind of a very, very fundamental question in what we're talking about. And this is basically the reason why I brought the whole thing here, because I want to talk to you about what we know today about single neurons that we didn't know 20 years ago. Okay. So 20 years ago, we knew that when you take a neuron and you depolarize it to threshold, it would spike. And this is true also today. If you depolarize a neuron, it will spike. That's basically everything that we know. We know also that by definition, excitation increase the probability of spiking, and inhibition reduce the probability of spiking. That's the definition of excitation and inhibition. Okay. But this is very naive view of what excitation and inhibition does. So if you think of excitation just makes you closer to threshold, inhibition takes you, away from threshold. This is not really the whole story. So I will try to show you some things about that. Okay. Now we also saw now that the dynamic of the network has something to do with the effect of what makes a neuron spike. But I'll show you some things that you don't know about. Okay. And then it's important to know that this is not everything. So if you go here, most of the, 95% of the scientists, neuroscientists will say this is everything. Okay, excitation, inhibition, maybe some complicated interaction between excitation and inhibition that I'll talk about, and that's it. Okay, but now we have a lot of data to show that this is not the whole story. Other things can also make neurons spike. And not all the neurons are the same in the way that they are actually process their input. Okay, so like the next lectures are going to focus on these issues, and we'll take a break and then we'll come back to that.
זה הגיוני, גזרתי לפי הממוצע.
לא, אצל החצי קראתי הבדלה כזה.
עכשיו בוא נסתכל רגע על סיכון לגזרות של... אתה גוזר לפי, נכון? כן. סיכון לגזרות של X. לגזור את FE, לגזור את זה, תשימו GX, זה כך. עכשיו החלק של FX, ואני גוזר את GX, זה מה שרשום בעצם בכל זאת, כי גזרנו בכל זאת.
这步骤应该它可能还没上传，我不清楚，可能还没传吧。
אבל זה וקטור, זה וקטור, אחד שם אמור להיות דיפרנציאל, אחרת זה לא הגיוני.
כן, אתה אמור לקבל מטריצה.
יש לי מטריצה בצד שמאל, כאילו, בצד שמאל של הפלוס יש לי מטריצה.
כן, אני רואה. כן, אחד מהמטריצה
אפשר לטמצל את זה ולקבל מטריצה, לטמצל את זה ולקבל מספר. זה אנשים מקוקיים.
לא, מספר אתה לא יכול לקבל, כאילו לא שזה תלוי בה. אוקיי, זה לא מעניין.
כלומר, כשמחפשים את זה בתחתו?
כן. אבל אני מבין, כי אתה צריך לגזור לפי וקטור טרנספורט. צריך לגזור לפי אתה גוזר, פעם אחת, אתה לא יכול לגזור פעמיים. בגלל העניין הזה אם אני חושב ראשון שהייתה לפי פעם הבאה עוד 100 שנה
אבל ליגי אנחנו עושים הפסקות של טוב לב וואי, אני משחק לבוסים
הדבר הזה בטרנספורט
אבל ראיתי באחרים
לא, רוב המוזיקה לא עזית. מוזיקה ישראלית הגענו לו כמה שירים, כי הוא תפתח שם באמצע, פשוט עכשיו הוא יפתיגו לזה.
מה זה הכי גרוע?
כל החיבורים מפרקים וזה.
רגע, מה זה עמודה כפול? זה עמודה כפול
יש
שורה.
עולם גלובלי.
Not like fresh air, like more fresh than I think I can it can take brief.
התשובה הנכונה, תמיד תקשו מביאה sì, dopo che andavo un po' di finire الشغل اليوم يتوفي الشغل تيجوا ما تقدروا
Did you go Sunday? Yeah, did you go Sunday to Odessa? How is it? Yeah, and then?
כן, דיימן נתכנס
Yeah, I don't know, it was too stressful for me. Getting there, because you know, next week the dynamical processes start. I'll take it in 2 years. I thought I will study the articles on my own.
Okay, so just few really kind of, I'll do this part very quickly. Maybe we'll get back to it at the end of this section. But the question is like, okay, so someone asked me before, what are the evidence for this type of coding, for another type of coding? What would you do to answer this question? How would you address it? Now, you know, forget about all the models and all the theories, all this. I don't want to use bad words. What can we really do in order to try to answer this question? I mean, I try to convince you it's a fundamental question. What can we do to answer it?
You can somehow change the activity of neurons and see the kind of perceptual mistakes they make, but it was possible experimentally to do that.
Okay, it's a good direction. I'll mention it in a second.
Change the parameters and see what happens.
What do you mean? Change the parameters of what?
If you have a model, then you can change the parameters either in your simulation in experiment.
Well, what is changing the parameters of the experiment? Obviously, we need to change something, right?
Capacity of the neurons, number of ion channels. I don't know.
Okay.
Change the stimulus parametrically.
Yeah, okay, so this is more the traditional way. So what people were doing, first of all, in order to see that the neurons are representing information in the first place, is we're looking for receptive fields, we're looking for things that are making the neurons spike. Okay, and then what we can do is, you know, play with these parameters and see what changes in the output of the neurons or the network, and then try to connect these two things. So this is a classical direction, and people took it a little bit kind of more. So this is what I wanted to show you, a little bit in a, you know, some interesting ways. For example, one of the methods by Victor and Purpura, started by Victor and Purpura in 1996, it was inspired from DNA sequences. When you have two genes and you're trying to compare, I don't know, a gene of a mutant to the original wild type, or between parents and offspring. So what you can do is look at what we call the editing distance. How many things you need to change in one sequence in order to get to the second sequence. Okay, so for example, one thing you can do is like present a visual stimulus, record the spikes, then present the visual stimulus again, record again the spikes, and then see how much do you need to change one spike train in order to get to the other spike train. And this gives you some measure of the variability that you have between different repetitions. Okay, and you can do it again and again and again, and you can compare it between different cases. And so this is an interesting algorithm if you want to look at it, and you can also get some interesting measurement out of it. Another way to do that was by measuring entropies. So really using information theory to quantify how much information there is in the output of the spikes on the original stimulus. So one of the original works, what they were doing is they were taking a fly, okay, and recording from a specific neuron in the fly eye, okay, and then moving the fly or moving the world, it doesn't really matter, and this neuron was sensitive to movement of the world. So you get spikes, and the spike correlate with the movement of the world. Okay, so you can see it here. The stimulus. This is the velocity of the changing of the world, and these are the spikes that you get. Okay, now you can do two things. One of them is you can just give a very, very, very long stimulus, okay, and record all the spikes. And the second thing that you can do is take a piece of movement and repeat it many times, and then every time you get a slightly different spike train. Okay, so this is for the first presentation, second presentation, third presentation. And then you can say, okay, so if I give the same stimulation and I get some variability, this variability is noise, because I gave exactly the same input and the neuron was doing something else. This is a noise. Okay, and from the very long presentation that I get the variability of the spike train. So by using these two measurements, I can actually calculate how much information there is in the spike trains on the stimulus, and I can play by looking at different bins and find what the relevant bin in order to get some information out of the output. So this is just kind of, I'm not going to talk to you about these things too much, and I'll get back to that maybe at the end to show you that there are also some evidence, first of all, for thin fire chains in certain cases, and also some evidence for where noise can actually be beneficial. But we'll get to all that at the end. Okay, so now I want to switch the topic and move to switch to the single neurons. And I'll also skip the first few slides because I talked about that. Okay, so I want to start with talk in a little bit more detail about single neurons and their physiology, things that we know about their physiology. So a lot of that you already know from the course of Inbal, and some of it Dan talked to you about in the previous course. But I just want to show you that neurons, very differently from what people are doing in models, they have to face a lot of problems that they have to overcome just in order to do the function which we don't know what it is. Okay, for example, one of the problems is the length of axons. Okay, so when you imagine a neuron in the cortex that sends axons all the way down to the spinal cord, this could be a meter long axon, and this neuron, all the things that happen at the terminal of this axon, some of the molecules have to be synthesized in the soma of the neuron. So a lot of what the neuron is doing is just kind of maintaining its being a cell, okay? And even from cell biology point of view, these cells are very, very sophisticated compared to many other cells that we have in the body, just because they have to solve all these problems of transferring things to the terminal, transferring things to the dendrites, find the right contacts, whom to talk to, whom not to talk to. Even the organization of the node of Ranvier, where the spikes kind of jumps between the sections of the myelin, you have to bring all the ion channels and organize them in a certain way. It's not random. The structure in every resolution that you will look at, and it's really difficult to even conceptualize this thing, because this is 20 micron and this could be meter. Okay, so just the order of magnitudes are huge. Okay. So let's talk about the membrane first. Just remind you the really, really basic stuff. So the membrane is this lipid bilayer, and in between them, there are all these proteins that are the ion channels, and also many other proteins. And this the function of this protein is to maintain this difference in concentration of certain ions between the inside and the outside of the membrane. What's inside? What's the high concentration? Schlegan, potassium. And the outside? Sodium. And what about calcium? How much more?
Much more.
So there is more potassium than inside than outside, more sodium outside than inside, and calcium, there's a lot of inside, but it's not free. It's bound to protein. So the proportion, if you look at calcium, is many more order of magnitude compared to sodium and potassium. Okay. So how do we study them? We study them with patch clamp, and I think I showed you some of these things when we talked about in the method course. This idea is that you can take an electrode, suck a little bit of the membrane in, and then isolate. So basically the membrane sticks to the glass, and this piece inside the pipette is isolated from the outside wall. So every current that flows through the pipette can only flow through this protein. And when you look at these proteins, they have a certain structure. They're usually made out of different proteins that couple together. So this is one protein, and it has different subunits. Okay, and so a channel can be made out of 4 copies of that thing that are combining together. Okay, and these subunits have some function. For example, this subunit 4 is usually the voltage sensor. So if it's a voltage-dependent ion channel, subunit 4 will have some polarization of its structure, and then when the membrane voltage will change, this thing will move inside the lipid bilayer, and this movement will force something to happen in the channel and let ion get in or not. Okay, so around 2002, I think. Okay, so before that, using this voltage clamp method, people could characterize these channels by doing single channel recording. And single channel recording was very difficult, mainly because the noise. You have to record really, really small currents. So you have to get very, very clean recordings. And this is an example what happens when you do a single channel recording. You hold the membrane potential at minus 80 millivolts, and then you force the membrane voltage to be minus 40. So you start at minus 80, you jump to minus 40, and suddenly you see these currents flowing through your pipette. So each one of these lines is a trial, and you see that you have different kind of occurrence of this current at different times and different amplitudes. If you sum up all of them, you take the average current, you get something like that. So on average, the current behaves like, the current as a function of voltage behaves like that. So when you jump from minus 80 to minus 40, you'll have a transient voltage, a transient current coming into the membrane and then closing down. Okay, so this is the sodium current, inward current that closed down because of inactivation. Okay. So this is average over many repetitions, but you see that each one of these is kind of random. So people try to fit all kinds of model to that. We're not into modeling right now. But what I want to show you is basically why Rod McKinnon, who did this, found the structure of the fat channel, why actually this was so important and so not obvious. So I'm not going to go into details of this calculation, but what you can do is kind of, you can assume what is the size of the pore of the channel, what the size of the hole in the middle of the channel, and then ask yourself how many ions are going through. So this is a potassium channel. How many ions are going through this channel when you open it? Basically, what is the current? The current is a flux of ion or charge. So you can basically count how many ions needs to go through the channel to account for measurement like we saw here. So if this is sodium, but assume you are now recording potassium, you can calculate how many ions just from this measurement. Okay. And the current, if you get something like 10 picoamp, okay, this is 6 times 100 million ions per second. That's the number of ions. That's the current that you measure through the single channel when it opens. Okay, but you can also calculate it, assuming that you know what the size of the hole and what the size of the ion. And you see that this channel achieves almost, 1/10 of the, it's not tenths, it's almost the maximum that you can imagine. So if it was a hole, and you would just count how many ions flow through the channel, it behaves like a hole. Everything that can flow just by the diffusion flows through the channel. So this looks kind of simple, right? So you have a channel, you have a hole, the ion flows, everything is according to plan. No, so this is a channel, imagine it's a channel without voltage dependence.
No, about there's also electrical force involved, not only the expression.
No, you take it into account when you do this calculation. So this is not the point. The point is that when you remember that the sodium ion is actually larger than the potassium ion is larger than the sodium ion. So if it would be just kind of moving through, then the sodium should rush at more or less the same rate or even higher rate. Okay, but it's not like that. The channel is highly selective. So if you looked at the selectivity, you get one sodium per every 10,000 potassium ions. So this doesn't happen. It's not like 1 for 1, it's completely selective. Only potassium goes through, but it goes at a rate as if it goes smoothly. Okay? This is very mysterious. How does the channel do it? Okay? And for that, he got the Nobel Prize. And I'll just do it in a very short way because it's quite complicated. But the idea is that the channel has a very specific, these proteins have very specific structure when you look at the pore, at the inside of the pore. And these points in the pore, what they do is they imitate water molecules. So when you have a potassium ion in the water, it's surrounded by water molecules. Okay? So when it arrives into the pore, these two parts imitate the water molecule. The water molecule is basically detached, and it kind of, the ion sits here as if it's surrounded by two water molecules and two actually imagined water molecules. Okay? Then the next ion pushes it in, and now it's surrounded only by the protein, but it feels as if it is surrounded by the water molecule. And this is what lets the ion go through the whole way inside the pore. Okay, now, if you try to do it with a sodium ion, it's too small and it doesn't feel comfortable in that sense. These are too far away from the sodium ion, and it doesn't go through. Okay. Now, some ion channels that have only, half of this structure. Okay. A certain ion channel that I happen to like and work on has like a twist here, and these two are actually facing that side. Okay. And then this channel becomes much less selective for potassium and sodium, and they kind of almost transfer them on an equal basis. Okay, so you need to have it like 4 times in order to get this selectivity. Otherwise, the sodium kind of, just by the bump of the other ions, can go through. Okay, so this is more or less what it looks like. So this is a single ion in the middle of the pore. And the pore has this structure that when the voltage changes, these, you see these points here kind of change the conformation and let the channel goes in. Okay, so again, why is it important? It's important because these are the basic blocks that make this makes neurons. It's also extremely important for other things, like if you want to find drugs against specific ion channels, if you want to understand the limits that you have in the system, and because eventually we try to go from these ion channels to the current that dictates the behavior of the channel of the neuron. And neuron has tons of these currents. I mean, there are hundreds of ion channels, and each neuron has basically a repertoire of which channel it chooses to express. And it turns out that they are doing it in different ways, and this is what makes them different neurons. Okay, so if you are a physicist, you know only 2 types of neurons, excitatory and inhibitory. Okay, and also this is only if you are dealing with neuroscience, because like in computer science, you can kind of also change the synaptic weights to be positive and negative. No one cares about that at all. Okay, but in real life, there are many, many types of neurons, and what makes one neuron different from another, one of the things is the composition of the current that it has. And I will try to show you that it makes some differences. So this is kind of some kind of representation of how do you represent the properties of a current. So for example, the sodium channel that we saw before is a current that when you take the voltage and step upward the voltage, the current opens, injects current into the neuron. So this is like when we get the current down, this means an inward current, a positive ion that goes into the membrane. And then it inactivates after some time. So this is a transient sodium current that you know that makes the action potential. But for example, there's another type of action of sodium current that is called persistent sodium. And persistent sodium is a current that when you depolarize the membrane, you don't need to cross threshold to activate it. Okay, even if this is a threshold, you can make a small jump in voltage. The current opens and it inactivates, but you see that it doesn't inactivate to zero. So it keeps being open. So you have constantly flow of current into the neuron. This is called persistent sodium. So what would the current like that do to a neuron?
The pressure will
It will change the threshold in a way, but what? Why higher? Lower in a way, right? Because
it's harder to make.
no, once you depolarize the membrane a little bit, it kind of helps you go over the threshold because it injects positive current.
But it makes some of the sodium ion channels being inactivated, and then you have less.
No. So you have these sodium channels that makes the spike, and then you have other ion channels, which is called persistent sodium. We don't know actually that there are different sodium channels, but assume for a second that they are different.
Still, you have depolarization instead, and if it's long depolarization,
It inactivates.
Inactivated, and then you have less.
Okay, so it's a good point. So it's a good point. Okay, so it's a good point. So and it helps me explain to you what I wanted to say in a way. Okay, so one way to look at it is, a small EPSP is actually going to be boosted because it makes a small depolarization, and then the cell opens its own ion channels and depolarizes even more. Okay, so this current, you can think about it as boosting the input. It makes the input larger than it was to begin with. Okay, that's one option to look at it. But the other option is indeed, because it kind of maintains the depolarization for a long time, this will inactivate those ion channels, because you know that when you depolarize, it inactivates the channels, and then you'll have less of them available in order to generate a spike. This can change the threshold. So the idea here is indeed that it's really difficult to say for each current what it does, what's the function of the current. The electrophysiologist likes to do that. Say, okay, this spike, this current is making this current is making a spike, this current closes the spike, this current is making burst, this current is doing something else. The truth is that these currents are complicated, and you need to consider them all together in order to understand what the system does. I'll show you in an example in a second of how people are addressing that. Okay, and you have so if we go back to this picture, you have also other kind of currents. For example, potassium currents, which are outward currents. So if you depolarize the membrane, you have a potassium. This is called IA. A current that goes outside, so potassium basically goes outside the membrane and then inactivate, but you have also others that don't inactivate. So if you depolarize, they hyperpolarize the membrane potential because they are taking potassium out. So the combination of all these, and you have here all these names are the names of the toxins or the blockers that enable us to close a specific current or not when we do experiment. So for example, TTX blocks sodium channels, and TEA, as you know, blocks the potassium channel that delayed rectified potassium current. But there are others like 4AP in high concentration, low concentration, that blocks different channels. Okay. All this field is called pharmacology. So how do you use certain drugs in order to block certain currents? So one book that I highly recommend to you is by Izikevich. It's called Dynamical System in Neuroscience, and the idea there is to take these current as equations. and make models of neurons. And I don't know if I have. Let me see if I have that. No, I don't. Okay, so you can. I'm sorry. But it turns out that on one hand it looks like you have infinite number of possibilities, but the geometry of this problem is less complicated than you might expect. So you have certain type of firing of neurons that you can expect, and you can achieve them in different way by different combination of these currents. But you can also classify them in things that are unexpected. So, for example, one classification that you might see is what's called type 1 and type 2 neurons. Do you know what these are? Anyone heard about them? So what's the difference between type 1 and type 2? Okay, so what does, someone who records from a neuron, what the first thing that they always do? What did we say? That depolarizing a neuron makes it spike. Okay, so usually when you go into a neuron, you have something that looks like that. You have a protocol that injects some hyperpolarizing current, and then no current, and then some depolarizing current, and more depolarizing current. And what you would see, if you record from this neuron, the voltage is that, for a hyperpolarizing current, you see something like that, right? And then for no current, you'll see something like that. And then for depolarizing current, maybe you'll see something like that. And if you increase it more, suddenly you'll get a spike, another spike, something like that. Okay? So this is what a neuron would behave like. And then you can count, you can plot a graph that looks like that. So here would be, how much current I injected, and here would be how much spikes I got per unit time. So a firing rate of the neuron. Okay. So how does it, what does it usually look like? Usually you would, for a certain amount of currents, until here you don't get any spike, so it's 0. And then it can increase, right? So you can kind of say, if it's very simple, it would be something like that, linear curve. The more current you inject, the more current, the more spikes that you would get. Of course, at some point it has to go through some saturation because of what? Refractory period, right? You cannot go beyond some limit. What are the numbers here, more or less? When do we get to, what is the maximum firing rate that we can get out of, say, a pyramidal neuron in the cortex? 100 Hertz is difficult. But yeah, something like, 50. Okay? So this is way below, so the neuron stops increasing the firing rate already below, before you get to the refractory period. They just don't like to spike too much. And if you do inhibitory neurons, how fast can they spike? Fast spiking neurons, what we call fast spiking neurons. How fast? More or less? 70? Who say more? Let's do a bid. What? 200. Anyone more? 1,000 is hard. There are neurons that do 100, but yeah, it's around 250. Sometimes you can even go to 500. Okay, the fast spiking neurons. And there are some that can go even to 1,000, but these are rare. Okay. So fast spiking neurons are 200 to 500, and regular firing are around 50, but sometimes they fire bursts, and then inside the burst you can get 100 spikes per second or something like that. But only 2 or 3 spikes. Okay, but there are neurons that are funny. So they kind of you inject the current. You go to more or less threshold, and then instead of doing something like that, they jump. So there is like a minimum firing rate. So below this firing rate, you don't get any spike. And above this firing, you can increase it. But they don't start, they never start with one spike and then wait and then another spike. They either fire and then they give a train, or they don't. Okay, it turns out that this distinction between, so this is, for example, a Purkinje cell behaves like that. Purkinje cells in the cerebellum. Okay, and this distinction, you can actually find analog to it in terms of dynamical systems. So it's kind of a bifurcation, and there are two types. And if you get one type, you get type 1, and if you get the other one, it's type 2, and these are the only possibility. Okay, so this is kind of, for some of you, might be interesting. So this is like the simplest way that we can kind of look at neurons, how they integrate inputs that we just inject current and look what they do. And we have the Hodgkin-Huxley model, and we can also complicate it by looking at the fact that neurons are not a single compartment. So this would be something like the Hodgkin-Huxley model, where you have a single compartment with these different currents, but neurons are not single compartments. They have axons and they have dendrites. Okay? So this is what it will look like if you open, the book, and this is what will look like if you open the brain. Okay, neurons are more complicated. They have very long dendrites. They have very long axons, and this has effect. It turns out that the dendrites has effect on how the integration of the current is being integrated. So Dan talked to you last year on compartmental modeling, so I'm not going to go much into the idea, but you can kind of think of dendrites as kind of disconnect them into sections, and each section think about it as if it's like a single compartment, as if it's one neuron, but then they are connected with these resistors to make the shape of the whole dendritic tree. Okay? And because of that, we call it compartmental modeling, because each one of that is like a compartment, and we assume that it's independent of what happens in the other, except of this current between them. And just to make clearer the point that we have all kinds of neurons, this is like a slide that Bartlett Mell, the person that we'll talk about soon, has made just to illustrate this fact, basically. So you have very, very complicated neurons, like the cerebellar Purkinje cell, which has a very elaborate dendritic tree and receives something like 200 or 100,000 inputs. Okay, it's a huge number of inputs. And this is in the cerebellum. And in the cerebellum, unfortunately, we don't have it, but there are neurons which are very similar to these neurons, which are granular cells, and they have only 4 dendrites, very small dendrites, and they receive only 4 inputs. And these are like very close to each other. The Purkinje cell with 100,000 inputs, and the granular cells with 4 inputs. Okay, and then the Purkinje cell, for example, in the cerebellar cortex of rats, you can compare them to fish, and you see that there are things that are similar. Okay, for example, they're both planar. So if you look at them from the side, they look like 3-4 microns thick. Okay, but if you look at them from this side, like here, this could be like 300 microns. Okay, so these and these are very similar, but here you see that the dendrite is just like very, very long thing, and here it actually branches all the time. Okay, so there are differences in their shape. There are these neurons that I talked to you about in the nucleus laminaris of birds, where they get inputs from one side, from one ear and from other ear, and kind of do this coincidence detection. And there are neurons in the cortex and in the retina and in many, many shapes. And it's one option to look at this is just say, okay, it's just a matter of connectivity. These neurons need to connect to collect input from a certain point. These neurons need to collect input from different points, so they just change the shape of the dendrite. But the other way to look at it is that maybe because they are doing different things, they actually, take advantage of that fact that the dendrite looks different, and they actually use it in the way they integrate input. And this is what I will try to show you in the next talk. I'm going to skip that. I just want to close with basic things that we know from the things that Dan told you about. The passive properties of dendrites imposed on a neuron. So if dendrites would just be cables that transmit information in a passive way, and the ion channels, there's no ion channels on them, then except leak, then this is the picture that we know for modeling and also from experiments that exist in these neurons. So for example, one difference when you have dendrites is that if you position a synapse on the soma, okay, you get a much more depolarization in the soma than if you position exactly the same synapse on the dendrite. Okay, and you can see it in the following way. So if you look at the voltage locally, what happens when you inject the current here in the dendrite, you get a very large depolarization in the dendrite. But most of the current that is injected here is actually flowing away, and then the actual voltage that generated on the way has this profile of decay. So this is the amount of voltage, and this is the x-axis, the distance from the soma, if you want. And you see that by the time you get to the first branch, you get something like 10% of what you got locally in the dendrite. Okay, and this continues to decay, and when you arrive to the soma, you get much, much less depolarization in the soma than in the dendrite, and also compared to what you would have in the soma if you would put the synapse in the soma. On the other hand, the decay in the other direction is not symmetric. So for example, you can see it here. So you get 10% of the original by the time you get to the first branch, but then if you look at the tip of the other branch, you see that there is not much of a decay into the branch. And the reason is that when the current flows into the branch, it feels like something that is closed. It cannot flow freely. It has to go through the membrane, and when it goes through the membrane, it generates voltage difference. Okay, so also the decay from the soma into the dendrite is smaller than the decay from the dendrite to the soma. So we have asymmetry in the decay of voltage. And this is what we call steady state voltage. So we inject current and we keep it on all the time, and we see how it decays with distance. Okay, what happens when we have transient voltage? So if we have like a very short injection of current, so you see this is the EPSP in the soma, this is current corresponds to that. Okay, you see a fast transient in the soma, but the transient that will be generated at the dendrite, it changes shape of it. It's kind of becoming slower. Not only it's smaller, it's also slower. Why is it slower? Because when you inject the current in the dendrite, it kind of charges, on the way to the soma, it charges the membrane capacitance of the dendrite on the way, and then you have to wait for that charge to relax back. So this is kind of filtering out the signal, and instead of get like fast transient, you get very short, a very slow signal. This means that if you have many inputs coming onto the soma, you'll see something fluctuating. Okay? But if you put them on the tips of the dendrite, you would see just like something smoothing completely the fluctuating input. So for example, when we talked about the summation of inputs before, how can they generate this fluctuating current to generate this random-like output spike train? This would work if all the input would happen in the soma, but it wouldn't work if most of them will go to the dendrites and the dendrites are very big. Okay? So this is important to keep in mind, and I'll continue with that in the next class. All right? Okay.
זה נראה שזה עושה אותו דבר, לא? כן, הבעיה שלי שפה אני לא יכול להכניס את כל זה זה לא בדיוק וקטורים, זה כאילו, זה אמור לצאת הסקלה, זה יוצא הסקלה, אבל השאלה אם הסכום של הסקלה, אתה יודע, זה בדיוק מה שאני אקבל פה. ההנחה שלי שכן.
כן, כנראה שכן. אתה יכול לכתוב את QX, X טרנספוז Q לפי אינדקסים.
קיצור, נשמע שפשוט הפתרון הראשון שלי היה נכון.
אבל לך להבין את
אבל
נראה בסדר. אבל זה קשה לעשות את השטויות האלה.
אתה משחק את זה מספיק כאילו תכלס כמעט תמיד מספיק לדעת נגזרת של זה
That shows